Hello dear students, we are going to learn a topic in this class is characteristic feature of genetic code. Characteristic feature of genetic code. Genetic code under topic genetic code that discussed in earlier class. Okay, now the properties first. Genetic code is triplet. Genetic code is triplet. Especially in genetic code dictionary, it is clear that each amino acid coded by triplet codon. Suppose this is a U, 3 codons that present on mRNA code by amino acid phenylalanine. It is clear that. But before triplet concept, some other concept of given is called singlet codon concept. Singlet codon concept that are also not accepted because 4 codon only code 4 amino acids. Now further the doublet codon concepts come in existence. This is also not accepted because 4 multiplied by 4 total codon will be the 16 and they code only 16 amino acids. How this is possible? Okay, this is also flop because it is not accepted. Now the triplet codon concept was given by George Gamow. Which one is a George Gamow, and they give the concept of the triplet codon, and according to this 4 multiplied by 4 bases on there will be 64 and easily all 64 codon can code 20 amino acid in 20 amino acid in definite manner, because some codon are coded by the some amino acid coded by more than one codon. You can see the leucine can coded by the 6 codon. You can prepare a list of which amino acid is coded by how many codons. You can prepare a list here, because student this will be directly help you to remember and how amino acid can code many codons. Okay, now this one is the concept of George Gamow. Second important characteristic was the no overlapping. Some scientists believe that the genetic code will be overlap because on mRNA this is a U, G, C and a lot of AU, G, C, CU, okay, TA. So this one is a different basic codon. This one is, suppose this is a coded methionine. Okay. Now according to the overlapping concept, the codon will be overlap. Now if UGC code, then next codon will be UGC, and which one generally code? UGC generally code 16. Okay. No, but this concept was totally disapproved because codon is fixed and AUG is going to code which amino acid is also fixed. Okay, so the concept of no overlapping concept is totally disapproved because a codon is non-overlapping type. This one is a fixed codon, this one is a fixed codon, this one is a fixed codon. You can say that by deletion or addition of one additional base sequence, that is the shifting of frame either left hand side either right hand side is considered at the time of mutations. This will already learn you students in mutation, but in this class we also in this chapter we also learn about the different mutations of base substitution, base shift point and all this type of mutations. I left this topic at the time of discussion of chapter 5, so I discuss in this class. Now so this one is a no overlapping. Second is a no punctuation. Between codon no comma is given. If punctuation was present, then it is possibilities in case of genetic mutation no shifting of frame, because if punctuation is present, if one base deleted, it means this is not able to code protein. This UCA is not able to code protein. Further, this UTA can code protein. That's why there is no problematic effect that fall on the body, especially can code if protein is too long and made up of many amino acids, in that case alteration in single amino acid cannot affect a large scales. That's why this was the advantage of punctuation. But genetic code is not punctuation. No punctuation is found between the genetic codon. Why? Because it is seen that due to deletion or addition of bases, shifting of frame occur. Either deleted, they are shift right hand side. Either addition, they shift left hand side. So this type of phenomena is generally seen in case of genetic code. Now students, the third important point of the properties of genetic code is university. No overlapping, no punctuation. This is also second and third. Now fourth, no. Next important point is genetic code is universal. Genetic code is universal. What does it means? Genetic code applicable on all organisms. Applicable on all means from E. coli to human. From E. coli bacteria to human. It means that if UUU code phenylalanine in case of prokaryote, especially E. coli, then in human UUU code phenylalanine. Okay, so this one is a genetic code is a universal, means they shows universality. You can say the genetic code is universal, that is applicable on all type of organism ranging from prokaryote to eukaryote, from unicellular to multicellular, etc. Now the fifth one is a genetic code is degenerate. Okay, genetic code, this is also most important. Genetic code is degenerate. What does it means? It means that a single amino acid, okay, single amino acid coded by more than one codon, okay, a single amino acid is coded by more than one codon. This is a genetic code is a degenerate. Okay, so this one is a degeneracy. Generally it is considered. You can see the leucine can coded by 6 codon. This is a very important characteristic, okay, of genetic code, that a single codon is coded by, single amino acid is coded by more than one codon. But it is never seen that a single codon code more than one amino acid. It is not possible. Okay, so this is a fixed characteristic of the genetic code. Now next one is the code is ambiguous. Codon is ambiguous. Codon is non-ambiguous. Ambiguous, non-ambiguous. It means that ambiguous means clear. Ambiguous means clear. Suppose in mRNA AUU present. In absence of isoleucine, it code isoleucine. But in absence of isoleucine, it is never code AUU, never code other amino acids. Means each codon is fixed for a particular amino acid. If they are present, they can code. If not present, the protein synthesis will not occur. Okay, so this one is a codon is a non-ambiguous. Each codon is clear and fixed for a particular amino acid. It is also known by non-ambiguity of the codon. Non-ambiguity of the codon generally happen by the amino acids. Non-ambiguity of the codon means each codon is clear and fixed for a particular amino acid. Now the third is next characteristic, the codon having also initiation, especially AUG initiation codon. Next one is a initiation codon. It means that genetic code having one important codon is called AUG, especially this code for methionine. Especially if any mRNA going to code a protein, the first codon will be the AUG. If AUG absent, then it is a GUG. Okay, GUG. Okay, but methionine is the initial amino acids that code that start the synthesis of the protein. That start the synthesis of the proteins. Now this is initiation codon. At one is a nonsense codon. In genetic code in dictionary, 3 codon already explained you. 3 codon already explained you. It is called a nonsense codon. Nonsense codon are of 3 types. It's called UAA. Second is a UAG, and third is a UGA. These all 3 codons generally code no one amino acid. No amino acid is coded by this codon. No amino acid coded by this codons. Okay, students, nonsense codon are 3 types. You can see this one is a UAG, UAG, and third one is a UGA. These all 3 codon no have amino acids. If this codon come in mRNA chain, suppose mRNA having 1000 base pair, they can synthesize up 333 amino acids long proteins. And if any termination codon present in between, then the synthesis of proteins, synthesized protein will terminate. Synthesized protein will terminate in between. That's why the nature of the protein is not accurate as like that having complete amino acids. Okay, so you can say this one is a nonsense codon, especially terminate the amino acids, especially it is end of the mRNA protein translation. Now this is a nonsense codon. Nonsense, why nonsense? Nonsense codon because it is a terminate the amino acid. It is also called stop codon. This is it is also in another name it is also called a termination codon. Means it is a nonsense, stop or termination, both are all are same especially. Okay, now this one is all these are 8 that are genetic code. What will be the genetic code of organism? Students, I think you well learn about the genetic code. You can further proceed this. What is the need of to learn a genetic code, students? I want to explain you a relation between DNA, mRNA, proteins, codons. Okay, you know that the DNA if going to synthesize their protein, first DNA synthesize mRNA. DNA synthesize mRNA means DNA create a template. DNA create a template, especially a short polynucleotide chain of mRNA having length of, you have already learned at the time of discussion of mRNA. This mRNA comes outside from the nucleus and they present in cytoplasm attached with the ribosomal RNA. This having sequences of this of UGAC base sequences generally. This all they are sequences that are for particular amino acid that present that transcribed on mRNA. This mRNA is called transcript because the mRNA is synthesized by DNA by process of transcription. That mRNA having this coding sequences as UU, CU, CUA, this one is a coding sequences. And you know that if DNA transcribed mRNA by this coding sequences, it means that fix, means this coding sequences code these amino acids, means nature of protein is fixed at the time of transcription of mRNA formation. What nature will be the protein? Okay, so you can say that mRNA attached with the ribosome. Now tRNA taken one by one amino acids to the mRNA, transcribed there, especially not transcribed, translate there, because information comes on mRNA by DNA, given by DNA, especially they translate to the proteins. They create translation only, because transcript form already at the time of mRNA formation. So you can say just after the synthesis of whole aggregation of one by one amino acid, long polypeptide chain are formed on mRNA, and further the termination of protein synthesis stop on mRNA, and after some times the protein comes outside from that translation mechanism. mRNA comes outside, mRNA degraded again in nucleotides, and they move again into the nucleus for another transcription. Okay, so this one is the overall mechanism of transcription as translation. But here, because this topic explained you at the point of genetic code information, how the genetic code affect it, how the genetic code responsible for translation, how genetic code reveal the which codon, which triplet codon going to code which amino acid. This is very important. Student, you must learn this, because this is the very important. This codon you have prepared one time, learn again, learn 3 to 4 time revisions. You may easily learn all the amino acid coded by which codon, because you know the sequences. You only know the position: serine, valine, proline, threonine, and alanine. The 4 codons that code a particular amino acid. This is very important. Now, students, in next class, again we learn about chapters called transcription, synthesis of mRNA. Okay, I think this genetic code will help you to understand properly. Thank you.